Chapter 12, Part 4. In this part, I want to talk about the transmissive segment. First, you need to remember that the transmissive segment is the synaptic knob of the neuron. The main activity which happens in this segment is release of neurotransmitter from the synaptic vesicle. Before the arrival of the nerve signal, calcium pumps start to work in the plasma membrane of the synaptic knob. In this part, calcium ion can come into our cell. Then this calcium ion binds to the vesicles which are full of neurotransmitter. When the combination of calcium and vesicle take place, then exocytosis of these neurotransmitter and vesicles happen. In the next picture, you can see all the happenings. Here you can see that the nerve signal reach and come to the synaptic knob. When the nerve signal come here, the calcium ion can come to our cell by the calcium pump. You need to know that these calcium pump need to use ATP for bringing calcium inside the neuron. Then the calcium can attach with the vesicles which are full of neurotransmitter, and this combination can do exocytosis. You remember what is exocytosis? The vesicle attached to the cell membrane. This attachment again needs ATP, then the pore become open and the neurotransmitter released in the synaptic cleft, and after that it can attach to the postsynaptic cell. When it attaches to the postsynaptic cell or neuron, it can make the other signal in the next part. So, but in this part, we have transmitting of this nerve signal from one neuron to the next neuron or the next cell. Here in this picture, you can see what happened in your PowerPoint. If you check your PowerPoint, you can see the animation instead of this picture. This is the nerve signal which come to the end of our axon. Let me change the color here for you. Then the calcium pump start to work here, and the calcium ion can easily come to our cell. When the calcium ion come to our cell, it can attach to the vesicles which are full of neurotransmitter. This attachment can help exocytosis. In this picture, you can see that exocytosis take place and neurotransmitter are released in the synaptic cleft. Then they attach to the postsynaptic membrane and it can make the other signal in the next cell. This is about the transmissive segment, or the last part of our neuron. I will post these questions on the assignment folder for you, and then you can answer these questions for me. Now, I want to talk about the differences which we have between graded potential and action potential. First, you need to know that the location of the graded potential is the receptive segment of our neuron. But the action potential happen in the conductive segment. So this is the first differences between them. The next differences which we have between graded potential and action potential is this. The graded potential can be positive or it can be negative, but the action potential involves polarization and after that repolarization. The graded potential can make larger potential when the stimulation is stronger, but the action potential is the same. It means we have all none law for the action potential. It doesn't matter if our stimulation is weak or strong. When we reach the threshold level, which is negative 55, the action potential happens. When we don't reach the negative 55, we don't have any action potential. So this is the difference between graded and action potential. The other differences which we have between these two is this. The graded potential is very local and it can travel only in the short distance, but the action potential can propagate all the length of axon, and after that it can reach the synaptic knob. These are the differences which we have between graded potential and action potential. In this picture, you can see the difference again between graded and the action potential. In the first one, we have graded potential, and as you see here, it takes place in the receptive segment. As you see in this picture, here we have the strength of our stimulation. The strength is more in the beginning, and when we come down, the strength start to decrease and decrease. So this is the meaning of graded potential. But in the next two pictures, you can see the action potential. The action potential happen, as you see here, in the length of our axon in both pictures. One of our axon, the first one is unmyelinated and the second one is myelinated axon. In unmyelinated axon, as you see in this picture, we need to have the action potential or depolarization and repolarization in all the length of our axon. But in myelinated axon, you see that the stimulation can jump here. So in the part where we have the myelin sheet, our plasma membrane is insulated completely, so the ions cannot come inside and they cannot go out. Only in the nodes on here or in the neurofibril nodes, we can have depolarization and repolarization. And the movement and the speed of our nerve signal is much more faster than in unmyelinated fibers, as I talked about it before for you. So the conduction speed depends on axon diameter and the myelination of the axon. One of them is the diameter and the other one is myelination. First, you need to know that the thicker fibers can conduct faster than the thinner fibers. Thick axon offer less resistance to current flow down the axon. This is the result. And the myelinated fibers can conduct much more faster than unmyelinated. And as I told you, the information can jump in myelinated fibers, which we don't have it in unmyelinated fibers. We have three types of nerve fibers. A nerve fiber is an axon and its myelin sheet. Nerve fibers are classified into three major groups. One of them is group A, one is B, and the other one is C, based upon their nerve signal velocity. In the first group or group A of the nerve fibers, the velocity which we have is as fast as 150 meter per each second. These fibers have a large diameter. They are myelinated. Most of our somatic sensory neuron, which can extend from the sensory receptor to our central nervous system, and all somatic motor neurons extend from CNS to skeletal muscles are classified in this group. In the second group or group B, the nerve fibers can propagate nerve signals at approximately 15 meter per each second. And in group C, the nerve fibers can each propagate nerve signal at 1 meter per each second. In group B and group C, the nerve fibers are generally small in their diameter. They are unmyelinated or both of them. Visceral sensory and autonomic motor neuron, as well as a small somatic sensory neuron which extend from the receptor of the skin to the central nervous system, are included in group B and group C. frequency of action potential. The action potential are always propagated along the axon at the same amplitude. However, the action potential frequency can vary and is dependent upon the stimulation strength. When the stimulation strength increases, the frequency of action potential increases too. Consider the following. A brighter light initiates more nerve signal to be relayed from the eye along the optic nerve to the brain, and a loud sound can initiate more nerve signal to be relayed along the vestibulocochlear nerve, which I will talk about it later for you, from the inner ear to the brain. The brain then interprets the increased frequency of nerve signals as a more intense stimulation. In addition, frequency of action potential relayed along somatic motor neuron to a skeletal muscle increase muscle tension. So you need to know that let me color this for you. When the impulse amplitude is constant, the firing frequency varies with stimulation strength. When you have a stronger stimulation, you can have more response. When you have less stimulation, your response becomes less and less. And I will post these questions again on the assignment folder for you to answer them. Thank you.